So today I'll be discussing on fatty acid synthesis. Fatty acids are one of the important biomolecule and secondary source of energy. So de novo synthesis of fatty acid. Fatty acids are synthesized by an extra mitochondrial system. So the major site for synthesis of the fatty acids are liver, kidneys, adipose tissues, lactating mammary glands, lungs, etc. And acetyl coenzyme A is the immediate substrate for synthesis of free fatty acids, and end product is in the form of free fatty acids. So the cofactors requirement for the fatty acid synthesis are NADPH, ATPs, magnesium 2 plus ion, biotin, bicarbonate ion as a source of carbon dioxide. So the major fatty acid synthesized de novo is the palmitic acid, which is the carbon 16 carbon fatty acid. So what are the steps in fatty acid synthesis? The three major steps in the fatty acid synthesis are production. First is production of acetyl coenzyme A and NADPH, which are the basic raw material for synthesis of all types of fatty acids. Then second is conversion of acetyl coenzyme A to malonyl coenzyme A. And third is the reaction of fatty acid synthase complex. How these propagate, how these reaction propagate the synthesis of fatty acids. We will see one by one. So first we will talk about what are the sources of NADPH, because NADPH and acetyl coenzyme A are the basic raw material for synthesis of all types of fatty acids. So we will see here what are the sources of NADPH. So pentose phosphate pathway or HMP shunt pathway is the major source. Beside conversion of malate to pyruvate by malic acid, it is another source. And third is conversion of isocitrate to alpha ketoglutarate in the citric acid cycle. So these are the three important sources. So we can see it here, the synthesis of NADPH. So the malic enzyme, this is the malate and malate to pyruvate in the cytosol. It with the help of malic enzyme, there is a production of NADPH. So one important source. Second, the most important source is HMP shunt. HMP shunt is the major source for synthesis of NADPH and pentose sugars. So, so these NADPH are required for fatty acid synthesis. Then acetyl coenzyme A. Acetyl coenzyme A, the major sources for the synthesis of acetyl coenzyme A are pyruvate. From pyruvate to acetyl coenzyme A, then various amino acids like serine, alanine, etc. can be converted via pyruvate into the acetyl coenzyme A. Then fatty acid by beta oxidation, they convert up into the acetyl coenzyme A. So these are the various sources for synthesis of acetyl coenzyme. And then what is the utilization of acetyl coenzyme A? We better know. Like the major utilization is in the tricarboxylic acid cycle or Krebs cycle or citric acid cycle. Then they are also utilized in the synthesis of ketone bodies. These are also alternate source of energy. Once there is a lack of glucose sources for energy generation. Then third important role of the acetyl coenzyme A is in the synthesis of sterol and fatty acids. So this is the fate of acetyl coenzyme A. So we will see it here, the pyruvate to acetyl coenzyme A within the mitochondria. PDH complex is there. It is meant to convert this pyruvate to acetyl coenzyme A. Beside fatty acids, they are another source via beta oxidation. They generate a huge number of acetyl coenzyme A. Then amino acids, they also can be converted up into acetyl coenzyme A via transamination deamination reactions. And then these are the sources for generation of acetyl coenzyme. Then most of the time acetyl coenzyme A is generated in the mitochondria and from mitochondria to cytosol, because site of fatty acid synthesis is the cytosol, and acetyl coenzyme A production is majorly happens in the mitochondria. So from mitochondria to the cytosol, it cannot pass through the mitochondrial membranes. So there are alternate pathways or shuttle pathways for conveying or for transference of this acetyl coenzyme A right from mitochondria to the cytosol. So we can see it here, acetyl coenzyme A combines with oxaloacetate via the citrate synthase enzyme converts up into the citrate, and this is the first important reaction of Krebs cycle as well. So citrate being formed, it is further can be converted into isocitrate and further to glutarate, can propagate the Krebs cycle. Beside, depending upon once there is a need of production of fatty acid and it is in large quantities, so it is diverted for synthesis of acetyl coenzyme A, which will be further utilized for synthesis of fatty acid in the cytosol. So citrate can easily perish through the membranous barrier of mitochondria. It can easily pass through. So in the cytosol, this citrate can be converted up into the oxaloacetate, and via the via the enzyme citrate lyase. Here the coenzyme A is utilized and the acetyl coenzyme A is regenerated. So this is how there is an indirect, transfer of acetyl coenzyme A right from mitochondria to the cytosol via citrate. Citrate acts as a shuttle. So this acetyl coenzyme is not now available for synthesis of fatty acid. Further, oxaloacetate can be converted to malate via the malate dehydrogenase enzyme. Very well known reactions. It happens in the mitochondria as well as in the cytosol. This malate via the malic enzyme can be converted into pyruvate, and this pyruvate production leads us in the synthesis of NADPH. So we can see it from these two diagrams, understand nicely that how the production of NADPH and how the production of acetyl coenzyme A happens, and what are the different fates. So the two main enzyme of fatty acid synthesis are acetyl coenzyme A carboxylase. It is the initial and controlling step, rate limiting step of fatty acid synthesis, and second is the fatty acid synthase complex. So both these enzymes are multiple enzyme complexes, and the coenzymes and cofactors are biotin, NADPH, magnesium ion, and manganese. So we will see one by one further. So this is another clear view of the transference of acetyl coenzyme A right from mitochondria to a cytosol. So various shuttle mechanisms are involved here. So we call this shuttle mechanism as citrate malate shuttle. So I already told you in the previous slide, pyruvate to acetyl coenzyme in the mitochondria. Acetyl coenzyme A, if it is in excess, diverted to the synthesis of fatty acids in the cytoplasm. So some of the citrate which are going into the Krebs cycle, the citrate is coming out of the mitochondria. Here citrate is converting to oxaloacetate and further to malate, and this malate via the malic enzyme can be converted up into the pyruvate. So this pyruvate, this pyruvate can further enters the uh, mitochondria, right? And in the course of this conversion of citrate to oxaloacetate, there is a production of acetyl coenzyme. So this is how we have resources available in the cytoplasm, all set for production of fatty acids. So, production of acetyl coenzyme A and NADPH. So, a brief, more clear points about it. Acetyl coenzyme A is produced in the mitochondria by oxidation of pyruvate, already discussed, oxidation of fatty acids via beta oxidation, then degradation of some amino acids, and then from ketolysis or ketone bodies degradation. However, mitochondria is not permeable for acetyl coenzyme A, so alternate arrangements are made for the transfer of acetyl coenzyme A to cytosol in the form of citrate. So this all I have discussed, and these are the summary points about points about how these raw materials, acetyl coenzyme A and NADPH, are available in the cytoplasm for synthesis of fatty acids. Now, move on to the first step of fatty acid synthesis, that is the production or synthesis of malonyl coenzyme A. So acetyl coenzyme A is carboxylated to malonyl coenzyme A by acetyl coenzyme A carboxylase enzyme. So this is a one-step reaction. This is the acetyl coenzyme A, and this carboxylase means there is a addition of carbon dioxide. So carbon dioxide is available in the vicinity in the form of bicarbonate ions. So they can be exploited for production of malonyl coenzyme A. So only difference between the acetyl coenzyme A and the malonyl coenzyme A is that of this COO group. So which came from the came by the addition of carbon dioxide. So this is acetyl coenzyme A. One hydrogen is lost and replaced by the CO. The enzyme is acetyl coenzyme A carboxylase. So now two important substrates are available for further propagation of fatty acid synthesis. One is acetyl coenzyme itself, which is the source of this acetyl group, and second is the malonyl coenzyme A, which is an important source of two carbons units, which will be sequentially added one by one, and they lead to the chain lengthening in course of formation of fatty acids. So step one of fatty acid synthesis proper. So this is the fatty acid synthase complex. This is the monomer or one subunit. So it has two sides. One is the cysteine thiol group available, and second is the ACP, acyl carrier protein thiol groups are available. Both will be utilized for addition of aceto and malonyl groups. We will see how it happens. The first enzyme is acetyl coenzyme A transacylase. So once I was talking on fatty acid synthase complex, so 80 enzyme of the condensing unit that is now participating here in this step. So what will happen? It will lead to the addition of this aceto group, CH3CO group of the acetyl coenzyme A to the ACPS via the thiol grouping of SCP, this protein. Now COCH3 is added, and now COASH coenzyme A is released. Okay. So in the second step, this transfer of aceto group will takes place from here to cysteine thiol group so as to empty this site. So what is happening? The transfer of this group from here to this cysteine thiol group. So now this sulfhydryl group is empty for further acceptance of malonyl coenzyme A. Here there will be addition of malonyl coenzyme A. Component of malonyl coenzyme A will take place. The second enzyme is the malonyl transacylase enzyme. And this enzyme, what is it doing here? It is adding on the this malonyl coenzyme A. So coenzyme A is lost here, and via this thiol group attacks here this uh, nucleophilic carbon of the carbonyl, and it attaches itself. And with this, there is a subsequent loss of this SCOA in the form of coenzyme A. So now further, now both the thiol groups are added. One is added with the acetyl group, which is being derived from the acetyl coenzyme A, and the second is this addition of COCH2CO group, which is being derived from malonyl coenzyme A. So both sides are now occupied. Further, so we have seen the role of acetyl coenzyme A, AT and MT here. Next, further, this component is known as acyl malonyl adduct. So further, what happens here? There will be a transfer of this COCH3 group to this carbon because it has a carbonyl group which act as a labile, which is highly labile, which act as a leaving group. So this COCH3 now added here at this carbon. So with this transference, what will happen? Finally, the loss of carbon dioxide is there, and this entity is now completely added here. So the next enzyme is beta ketoacyl synthase or condensing enzyme. So the first unit, condensing enzyme domain, all three enzymes are utilized here, and finally we got the product ACPSCOCH2 and this COCH3 which was initially here, now it is added here. So CO carbonyl group is now converted to hydroxyl group in order to maintain the valency. Right. So this adduct is known as beta hydroxyacyl ACP. Further, what will happen? The now reduction unit domain enzymes will come into the action, and they start reducing this 4 carbon chain. So what will happen? First will be dehydratase enzyme. So beta hydroxyacyl dehydratase enzyme will work here, and that is going to remove this hydrogen OH group, and there is a hydrogen loss of hydrogen in the vicinity carbon, and this will lead to the loss of water. So with the loss of water, you will get a double bond here. So the 4 carbon entity or 4 carbon chain with a double bond. This is known as trans-enoyl ACP in oil. So it can tautomerize and it is stabilized. The system is stabilized. OH group is there, CH double bond on the alternate side. So they undergo no ketoacyl uh, tautomerism. So in the next step, this is the entity with the double bond trans enoyl ACP. Further, it undergo reduction by enoyl reductase ER enzyme of the reduction unit, and the NADP is required here for reduction. So it converts to NADP, and this double bond is now reduced, and now it has become saturated chain COCH2, CH2, CH2, CH3. So this is known as acyl ACP. Further, there will be a transfer of carbon chain to the cysteine thiol group. So you can see it here, cysteine thiol group is now empty. So after being reduced, this saturated chain along with the CO group, this 4 carbon chain is now transferred here. So there is a transference of this group, and now this kind of product we get, that is the acyl S enzyme. So this is the cysteine acyl group is added here, and now ACP thiol group is free. Again, all set to accept the another slot of malonyl coenzyme. So malonyl adduct is now going to be added as 2 carbon species. Further on this 2 carbon species, there will be addition of further 4 carbon species, loss of carbon dioxide, chain lengthening, then further reduction lead to the from 4 carbon to 6 carbon, and further cycle go on in the same pattern in all 6 steps will be repeated. So 6 more cycles of reaction are required for synthesis. If suppose we want to form palmitate has to be synthesized, which is the carbon 16. So six more such cycle has to be repeated. Further, once the enough length is acquired, so this is the 13, 14, and 15, 16 carbon length is now generated. Now it is grown enough. Now it is time to detach it. So the last domain, that is the releasing domain enzyme will activate, and that is thioesterase, and that what it will do, it will cut or splice from this ACP thiol sulfur. So this sulfur carbon bond is being broken up with the help of this thioesterase. So free fatty acid, palmitic acid is now freed. And this enzyme is also freed, and it is now all set to accept the another slots of acetyl coenzyme and malonyl coenzyme add on it, and further it can repeat the cycle and resynthesize the a fatty acid chain. So this is how the fatty acid synthesis takes place, and predominant free fatty acids most of the time is your palmitic acid. Beside stearic acid, what is the 18 carbon fatty acid that can also be synthesized. So here, you can see it, this is the fatty acid synthase complex. Two sides, this is cysteine thiol group and this is the ACP thiol group. And here there is an aceto group is added. Here the malonyl adduct is now being added. And further, this group will be transferred here after being reduced. So from 2 carbon to 4 carbon. Further from another malonyl is added here. Then from 4 carbon to 6 carbon after being reduced. So this is how the chain grows and finally it acquires the sufficient length, and finally the fatty acid of appropriate length is being formed and gets freed. So this is the overall scenario of synthesis of fatty acid. So for formation of palmitic acid, this is the complete summary reaction. Like 7-acetyl coenzyme A with the help of 7 carbon dioxide and 7 ATPs are being utilized. So 7 malonyl coenzyme A are produced, 7 ADPs, and phosphoryl groups are released. Now, regulation of fatty acid synthesis. So, two types of control mechanism regulate the fatty acid synthesis. One is the short-term control mechanism, and second is the long-term control mechanism. And this for the concentration of citrate in the cytosol is the most important short-term regulator, immediate regulator, because citrate stimulate the acetyl coenzyme A carboxylase, which is the enzyme meant to convert the acetyl coenzyme A to malonyl coenzyme A. So, which catalyzes the formation of malonyl coenzyme A, that is the rate limiting step. So, direct impact of citrate onto the acetyl coenzyme carboxylase, right? Then the level of citrate is high. If the level of citrate is high, so both acetyl coenzyme A, ATP are when both acetyl coenzyme A and ATP are abundant, so effect of citrate on carboxylase is opposed by palmityl coenzyme A. So palmityl coenzyme A through feedback regulation will oppose the uh, oppose the action of citrate on carboxylase. So palmityl coenzyme A also inhibits the transfer of citrate from mitochondria to cytosol, and also the 6-GPD, that is the main enzyme of HMP shunt, which generates the NADPH. We'll see in the subsequent slide. Then allosteric regulation. So activity of acetyl coenzyme carboxylase controlled by the phosphorylation and dephosphorylation process. In the phosphorylated form, the enzyme is inactive, while in the dephosphorylated state, the enzyme is active. Then glucagon and epinephrine, they stimulate the phosphorylation process. Insulin stimulate the dephosphorylated state. Means insulin always prefers the fat synthesis or lipogenesis, while the glucagon and epinephrine, they oppose the synthesis of fatty acid. So insulin is lipogenic, while these are glucagon and epinephrine are lipolytic and hormones. Here there is a more clear account of allosteric regulation. So this glucose, like citrate via pyruvate and acetyl coenzyme A, citrate to acetyl coenzyme A. Acetyl coenzyme A is now available for synthesis of fatty acids. So acetyl to malonyl coenzyme A, malonyl to palmitate via the fatty acid synthesis steps. Then here the NADPH being required, and further this palmityl coenzyme or palmitic acid, if it is in excess amount or in the sufficient quantities, it will through feedback regulation will inhibit the this glucose 6-phosphate pyruvate dehydrogenase. So this enzyme is being inhibited by the excess amount of palmityl coenzyme. So thereby this suppresses or inhibits the HMP shunt or pentose phosphate shunt. So this pathway is, if it is inhibited, there will not be production of NADPS. So this pathway of synthesis of fatty acid will be checked or stopped. Secondarily, excess amount of palmitoyl acid or coenzyme A will inhibit the action of acetyl coenzyme A carboxylase enzyme, which is in the active form in the phosphorylated state, active in the dephosphorylated state. Sorry. So this is the dephosphorylated active enzyme, and this is being inhibited by excess amount of palmitic acid. Further, it will be converted up into immediately after being inhibited by this palmitic acid. It is also to be converted to inactive form, that is the acetyl coenzyme A carboxylase, which is in the phosphorylated state. Again, once the concentration of is this palmitic acid is low, then once there is a requirement of synthesis of palmitoyl coenzyme A, so it gets dephosphorylated again via the phosphatase enzyme, and insulin is acting as an stimulator of this enzyme at that time. So it starts synthesizing palmitic acid via activation of acetyl coenzyme A carboxylase enzyme, which is in the dephosphorylated state. Correct? So, and the long-term control mechanism, it exerts its effect slowly by it is by induction and repression of enzyme synthesis at the level of gene. So this involves the changes in the gene expression which controls the rate of synthesis of these enzyme. So production of enzyme of fatty acid synthesis is stimulated in the liver when the carbohydrate and ATP are available. Once they are in the abundant quantity, so beside production of energy, the excessive amount is now diverted for the synthesis of fatty acid under the influence of insulin hormone. So, and it is decreased during the starvation and diabetes. At that time, the hormones are glucagon and epinephrine. They stimulate the lipolysis. Then high carbohydrate diet, all the enzymes of fatty acid synthesis are stimulated because excess carbohydrate to be converted to fats and stored. And the high fat diet and starvation and diabetes mellitus, all these inhibit all the enzymes of fatty acid synthesis in the nutshell. So this is the summary of all the regulation mechanism through short term and through long term control.